Hi students, welcome to BIOL-11 week 5. We're going to cover the process of cell division, two different processes. One is called mitosis and the other is meiosis. So just a quick review of everything that we now know about cells and parts of the cell that will be relevant for this. So animal cells and plant cells are different. They have different structures. So important for this lesson is that animal cells do not have a cell wall, but they do possess centrioles, whereas plant cells have a cell wall, but they don't have centrioles. We've also covered cell theory, which is that all living organisms are composed of cells. Cells are the basic unit of structure in all organisms, and all cells arise from other cells. This is important because this basically means that cells must divide in order to reproduce. So this process occurs in a cycle aptly named the cell cycle. Cell division, otherwise known as mitosis, is a fundamental process required for all life to exist. Not only do cells need to reproduce in order to generate other organisms, the way that bacteria will reproduce, but our cells also need to reproduce in order to regenerate our bodies. So for example, our bodies are constantly shedding skin cells every day. And if these cells weren't replaced, we would no longer have any skin, which obviously would not be good. So these cells are replaced by cell division. Now, the cell isn't constantly dividing, and these cell cycles actually differ depending on the cell type. Some are more rapidly dividing and some are not. Cells that constantly divide is basically the disease known as cancer. Cancer is just unrestricted cell growth and division. So there's a schedule in the cell cycle in which several important events happen. So in general, cells spend most of their time in the phase known as interphase. This is a state in which the cell has a nucleus, a visible nucleus. It remains intact, so it's easily visible by the microscope, and there are normal physiological processes going on. But the cell is also preparing for the next mitotic phase, in which the cell will actively divide. So we have three different phases of interphase. The first is known as G1, and this is when all the cellular contents, like the organelles, are duplicated. This does not include the chromosomes. This next phase is referred to S phase, which is named for synthesis of new chromosomes. So this is actually when the chromosomes are replicated. Each of the chromosomes are duplicated, 46 in our case. The next phase is G2, which is an important step of the cell cycle in which the cell double checks all of these duplicated chromosomes for any errors in replication, meaning was there a mistake in replicating the genetic code? This could result in a mutation. So the cell can use this time to fix these errors. And many cancer cells actually have mutations that either eliminate or impede this important double-checking phase, which then allows them to divide unchecked and often divide with errors, leading to accumulated mutations. All right, so S phase is very important. Every cell needs a complete set of chromosomes in order to survive independently. This image that I'm showing you is known as a karyotype. You can see each pair of the 23 sets of human chromosomes, 46 in total. If you look at this and think about what you might know about genetics, see if you can figure out the sex of this individual. And we will come back to this next week when we talk about human genetics. All right, so the newly duplicated chromosomes are referred to as sister chromatids. Each sister pair is connected by the centromere, and this allows these chromosomes to remain tethered to one another as they travel all over the cell, getting ready for the cell to package one set for each of the new daughter cells. You'll see that this becomes important during metaphase, which we'll talk about in a few minutes. So finally, the last stage of the cell cycle is known as the mitotic phase, in which cell division is actually occurring. The duplicated chromosomes line up in the middle of the cell. Then long strands, known as spindle fibers, attach to these chromosomes. They tether the chromosomes to the centromeres at a structure known as the kinetochore on the chromosomes. These fibers then pull the sister chromatids apart and away from the middle of the cell toward the poles, either side of the cell. The centrioles are located at the poles of the cell, and they're surrounded by a structure known as the aster, which basically is all the spindle fibers extending from the centrosome, and this actually resembles a star. Aster is the Latin root for star. All right, and since we're biologists, we love to divide and categorize. We also divide mitosis into different phases depending on the important events in this process. So I use a mnemonic device to remember the events in the phases of mitosis. So prophase is the first phase. This is where the chromosomes pair up and the nuclear membrane begins to degrade. The metaphase is when the chromosomes line up at the metaphase plate in the middle of the cell and the nuclear membrane is fully gone. During anaphase, the sister chromosomes are pulled apart, and then during telophase, the nuclear membrane begins to reform and cytokinesis begins. So cytoplasm is divided along with the organelles, which are duplicated during interphase, and the two new daughter cells during cytokinesis. This is very obvious to see under a microscope because you can start to see the formation of the cleavage furrow where the membrane actually starts to pinch in. And this is directed by a contractile ring composed of actin, and this physically actually just pinches off the membrane, similar as you would think of a sausage being pinched off. So how do plant cells divide? We've been talking about animal cells, but as we discussed in the beginning of the video, plant cells also have a cell wall that they're going to need to deal with. So mitosis progresses very much the same way as in animal cells, with the replication of chromosomes and organelles. But then these cells have to account for these other differences. Plant cells also do not have centrioles or asters, but they do have the spindle fibers which connect to and separate the chromosomes. During telophase, the cell plate will form across the middle of the cell, and this is composed of vesicles originating from the Golgi apparatus. And you can see this in the mitotic telophase, where you actually see that cell wall reforming across the middle of the plant cell. All right, so meiosis is a specialized version of cell division. And instead of creating two identical cells, it creates specialized sex cells, which are known as the gametes, eggs and sperm. These specialized cells combine during sexual reproduction to create a zygote. And since every individual cell must have the same number of chromosomes, a cell must reduce the number of chromosomes by half. Otherwise, when the egg meets the sperm, you could have a cell with 92 chromosomes, which would also be known as a dead cell. The number of sets of chromosomes in a cell is known as ploidy. We have 23 pairs of chromosomes, so meaning that we have a set of 2 for each of our 23 chromosomes. And if you go back to the karyotype, you can actually visualize this. So we're diploid organisms, and our gametes only have 1 set of chromosomes each. This is referred to as, they're referred to as haploid cells. So how does a cell accomplish this? Basically, the cell completes 2 cycles of cell division, one after another, except after the first cycle of cell division, instead of again going through another S phase in which the chromosomes are duplicated, it moves directly into another phase of division to reduce the chromosome number by half. We call these stages meiosis 1 and meiosis 2. So when we're discussing ploidy, we often denote it using a lowercase n. A diploid cell is represented by 2n, and this contains pairs of homologous chromosomes. After meiosis, the haploid daughter cells are denoted by an n, a lowercase n. And a pair of homologous chromosomes during cell division is referred to as a tetrad. So meiosis 1 is also broken down into prophase, metaphase, anaphase, and telophase. During prophase 1, the homologous chromosomes again condense and pair up. This is where synapsis occurs, and this is where chromosomes will cross over. And I'll talk about this a little bit more in a few minutes. These homologs align at the middle of the cell during metaphase 1, and then are separated during anaphase 1. And then there's the first phase of cytokinesis, which is called as interkinesis, takes place during telophase 1. After interkinesis, the cell immediately undergoes another cycle of division without replicating any chromosomes. The sister chromatids pair up during prophase 2, line up in the middle of the cell in metaphase 2, and are then separated in anaphase 2. In telophase 2, the nuclei reform in the daughter cells, and the final round of cytokinesis occurs. Therefore, from 1 2n parent cell, you now have 4 haploid gametes. So crossing over during meiosis is a very important driver of variation in biology. During that synapsis that I mentioned in prophase 1, in which the chromosomes can actually exchange genetic material, basically results that we have 4 unique daughter gametes from one round of meiosis. This includes 2 recombinant chromosomes, meaning the chromosomes that undergo crossing over, and then the parental chromosomes. So during crossing over, the DNA strand, the actual chromosome, is breaking and reforming bonds with the other chromosome. So this results in these recombinant and non-recombinant chromatids, which you can see in the final panel of the image. As you see that these chromosomes have exchange material. So this variation means that we can have many different possible combinations of genetic material during sexual reproduction. This is a very, very basic explanation about why siblings can look so different yet have the same parents. This means that you and your sibling can have the same parents, but you might not necessarily be getting the same set of chromosomes. All right, so the human life cycle overall involves many different mitotic and meiotic events. So mitosis is required throughout the life cycle for growth and development, the replacement of old and damaged cells, and all of these somatic cells, meaning those cells that make up everything else besides the gametes, are diploid. Only the gamete cells undergo meiosis and become haploid. So in gametogenesis, these are also very specialized cells. So it's not just that there's this division and we just have these cells that have these haploid qualities, but we also have, very specialized sex cells. The sperm cell and the egg cell look completely different. So this process of forming sperm, which occurs in the testes, is known as spermatogenesis. And then oogenesis, the production of eggs in the ovary, is oogenesis. So I'm including a lot of different phases in these processes just because I think it's important that you're aware of how specialized these processes are, but you don't need to know anything here, the spermatogonium, primary spermatocyte, or any of those. Just know that there are 2 distinct processes going on to create sperm and to create eggs. All right, so for today's lab exercises, it would have been microscope day. We would have been evaluating many different types of cells. So in lieu of that, I've posted some pages which include some very nice clear images of the samples that we would have been observing. So the onion root tip. I've also included some animal cells and I think some pollen grains to look at meiosis. So then also look at the activities I've posted. They're going to help your visual understanding of mitosis and meiosis, and I think especially crossing over. It's very helpful to see that in an animation. And as always, I have my office hours on Friday. Some people have come and talked to me, and I think it's been very helpful, and it was nice to see you. So if you have questions, please email me. You can set up an appointment or come to my office hours Fridays, 1 to 2 on Zoom. And the Zoom link is in my contact info on Blackboard. All right, have a great week, everyone. Set on the temperature.